right, so we are going to talk about mutations today. Like I said, you don't have to write anything down. You just have to know about it. At the end of the unit for our final, we're doing a project that has to do with mutations and genetics and all the stuff we've been learning. So just make sure you're paying attention to how mutations can happen. So I'm going to start with just an overview of a video. I think it's only like 3 minutes or 4 minutes. It's a TED Ed video, and then I'll get into the notes that are pretty short. I can't find my mouse. Hold on. Okay.
DNA in just one of your cells gets damaged tens of thousands of times per day. Multiply that by your body's 100 trillion or so cells, and you've got a quintillion DNA errors every day. And because DNA provides the blueprint for the proteins your cells need to function, damage causes serious problems, such as cancer. The errors come in different forms. Sometimes nucleotides, DNA's building blocks, get damaged. Other times, nucleotides get matched up incorrectly, causing mutations. And nicks in one or both strands can interfere with DNA replication, or even cause sections of DNA to get mixed up. Fortunately, your cells have ways of fixing most of these problems, most of the time. These repair pathways all rely on specialized enzymes. Different ones respond to different types of damage. One common error is base mismatches. Each nucleotide contains a base, and during DNA replication, the enzyme DNA polymerase is supposed to bring in the right partner to pair with every base on each template strand, adenine with thymine and guanine with cytosine. But about once every 100,000 additions, it makes a mistake. The enzyme catches most of these right away and cuts off a few nucleotides and replaces them with the correct ones. And just in case it missed a few, a second set of proteins comes behind it to check. If they find a mismatch, they cut out the incorrect nucleotide and replace it. This is called mismatch repair. Together, these two systems reduce the number of base mismatch errors to about 1 in 1 billion. But DNA can get damaged after replication too. Lots of different molecules can cause chemical changes to nucleotides. Some of these come from environmental exposure, like certain compounds in tobacco smoke. But others are molecules that are found in cells naturally, like hydrogen peroxide. Certain chemical changes are so common that they have specific enzymes assigned to reverse the damage. But the cell also has more general repair pathways. If just one base is damaged, it can usually be fixed by a process called base excision repair. One enzyme snips out the damaged base, and other enzymes come in to trim around the site and replace the nucleotides. UV light can cause damage that's a little harder to fix. Sometimes it causes two adjacent nucleotides to stick together, distorting the DNA's double helix shape. Damage like this requires a more complex process called nucleotide excision repair. A team of proteins removes a long strand of 24 or so nucleotides and replaces them with fresh ones. Very high-frequency radiation, like gamma rays and X-rays, cause a different kind of damage. They can actually sever one or both strands of the DNA backbone. Double-strand breaks are the most dangerous. Even one can cause cell death. The two most common pathways for repairing double-strand breaks are called homologous recombination and non-homologous end-joining. Homologous recombination uses an undamaged section of similar DNA as a template. Enzymes interlace the damaged and undamaged strands, get them to exchange sequences of nucleotides, and finally fill in the missing gaps to end up with two complete double-stranded segments. Non-homologous end-joining, on the other hand, doesn't rely on a template. Instead, a series of proteins trims off a few nucleotides and then fuses the broken ends back together. This process isn't as accurate. It can cause genes to get mixed up or moved around, but it's useful when sister DNA isn't available. Of course, changes to DNA aren't always bad. Beneficial mutations can allow a species to evolve, but most of the time, we want DNA to stay the same. Defects in DNA repair are associated with premature aging and many kinds of cancer. So if you're looking for a fountain of youth, it's already operating in your cells, billions and billions of times a day.
All right, so we are talking about mutations, and like the video said, it can actually help with evolution, things like that. So a mutation, the definition is the changing of a sequence of DNA that could create a variant and could get passed down to further generations. And if you want to use it in a sentence, mutations don't always result in a bad thing. So you might think like mutations are like can lead to cancer, like the video said, or can lead to like genetic disorders. But that's not always true. Sometimes it can actually be beneficial to organisms. The mutation is a permanent change in the cell's DNA. Mutations can occur by a spontaneous change in DNA replication, like how the video had that little guy that was putting on the pairs, or by a mutagen, which is just something that changes your DNA, like chemicals, radiation, ultraviolet radiation. So like x-rays, if you've ever had an x-ray, you have to have that heavy metal or like heavy, it is a type of metal, I don't know what type, like vest that they put on. It's to protect your cells from getting exposed to x-ray radiation. I don't know why this is back there, but there are some types of mutation. We only need to know a few of them, but this is how you can read them. So if we had the normal sequence would read the big fat cat ate the wet rat. So that would be like how the cells would read your DNA. But then if there's a substitution, it would change the sentence so that you can't even understand it, because it would change big into biz, and that just doesn't make sense. There's also another substitution that is nonsense. It would just like delete the end. Deletion is when they take out a letter from the DNA sequence. Insertion is when they add a letter to the DNA sequence. Duplication would obviously be copying. And then expanding would just be like repeating sections of the DNA. You guys don't have to memorize these, but I just did want to give you examples. And there's also diseases that are affiliated with all of these mutations. So achondroplasia, which is dwarfism, is a substitution. Muscular dystrophy is also a substitution. There's cystic fibrosis that is caused from a deletion. Crohn's disease is caused from an insertion into the DNA. I don't know how to say that, but this disease is caused by a duplication. And then Huntington's is from repeat sections of DNA. So one example of a mutation that I feel like is easier to understand and identify with, or you've probably heard of, is sickle cell anemia, which at the time when this was made, I'm not sure if it's true right now, but this PowerPoint was made like 2 or 3 years ago. It was the #1 genetic disease. The mutation type that sickle cell anemia is caused from is a base substitution. So instead of like an A, it would insert a T. The result is that the protein structure and like shape is altered or changed. And the effect is that the red blood cells are sickle shaped, causing blood clots due to the pointed shape, and anemia due to low volume of oxygen that the red blood cells could carry. So here is the, substitution. It would change this glutamine amino acid into this valine amino acid, and it overall changes the shape of the red blood cell from a circle disc to a sickle shape of a red blood cell. And we have another short video that talks about how this happens and what the kind of effects are, because your project, like I said at the end, for your, instead of having an exam like a test, will be about diseases like this. So I just wanted you to have an idea of what could happen if there's mutations in your DNA.
What shape are your cells? Squishy cylinders? Jagged zigzags? You probably don't think much about the bodies of these building blocks, but at the microscopic level, small changes can have huge consequences. And while some adaptations change these shapes for the better, others can spark a cascade of debilitating complications. This is the story of sickle cell disease. Sickle cell disease affects the red blood cells, which transport oxygen from the lungs to all the tissues in the body. To perform this vital task, red blood cells are filled with hemoglobin proteins to carry oxygen molecules. These proteins float independently inside the red blood cell's pliable donut-like shape. keeping the cells flexible enough to accommodate even the tiniest of blood vessels. But in sickle cell disease, a single genetic mutation alters the structure of hemoglobin. After releasing oxygen to tissues, these mutated proteins lock together into rigid rows. Rods of hemoglobin cause the cell to deform into a long, pointed sickle. These red blood cells are harder and stickier and no longer flow smoothly through the blood vessels. Sickled cells snag and pile up, sometimes blocking the vessel completely. This keeps oxygen from reaching a variety of cells, causing the wide range of symptoms experienced by people with sickle cell disease. Starting when they're less than a year old, patients suffer from repeated episodes of stabbing pain in oxygen-starved tissues. The location of the clogged vessel determines the specific symptoms experienced. A blockage in the spleen, part of the immune system, puts patients at risk for dangerous infections. A pileup in the lungs can produce fevers and difficulty breathing. A clog near the eye can cause vision problems and retinal detachment. And if the obstructed vessels supply the brain, the patient could even suffer a stroke. Worse still, sickled red blood cells also don't survive very long, just 10 or 20 days, versus a healthy cell's 4 months. This short lifespan means that patients live with a constantly depleted supply of red blood cells, a condition called sickle cell anemia. Perhaps what's most surprising about this malignant mutation is that it originally evolved as a beneficial adaptation. Researchers have been able to trace the origins of the sickle cell mutation to regions historically ravaged by a tropical disease called malaria. Spread by a parasite found in local mosquitoes, malaria uses red blood cells as incubators to spread quickly and lethally through the bloodstream. However, the same structural changes that turn red blood cells into roadblocks also make them more resistant to malaria. And if a child inherits a copy of the mutation from only one parent, there will be just enough abnormal hemoglobin to make life difficult for the malaria parasite, while most of their red blood cells retain their normal shape and function. In regions rife with this parasite, sickle cell mutation offered a serious evolutionary advantage. But as the adaptation flourished, it became clear that inheriting the mutation from both parents resulted in sickle cell anemia. Today, most people with sickle cell disease can trace their ancestry to a country where malaria is endemic. And this mutation still plays a key role in Africa, where more than 90% of malaria infections occur worldwide. Fortunately, as this adaptation thrives, our treatment for sickle cell continues to improve. For years, hydroxyurea was the only medication available to reduce the amount of sickling, blunting symptoms, and increasing life expectancy. Bone marrow transplantations offer a curative measure, but these procedures are complicated and often inaccessible. But promising new medications are intervening in novel ways, like keeping oxygen bonded to hemoglobin to prevent sickling, or reducing the stickiness of sickled cells. And the ability to edit DNA has raised the possibility of enabling stem cells to produce normal hemoglobin. As these tools become available in the areas most affected by malaria and sickle cell disease, we can improve the quality of life for more patients with this adverse adaptation.
So like the video said, sickle cell anemia or sickle cells arose from a mutation that was supposed to help species and humans adapt and not get malaria, a disease that could kill them and would not be good for you. But then sickle cell anemia also has its own side effects. So also, how do mutations affect gene regulation? So if there is a mutation, like if you have a mutation for the sickle cell disease, it can affect gene regulation, aka how genes show up on you. So like your traits, like hair color, eye color. Obviously, these are not just like our normal traits. They are things that might affect us or give us a disease. But if a mutation occurs, it can affect a gene by not turning it on or off. So like lactose intolerance, turning that gene on or off. So being able to digest milk and dairy or not being able to. It could not turn the correct gene on. So something in your body that needs to be on to regulate your blood or other things might not even be turned on if there's a mutation. And it also might change the timing of when a gene is turned on or off in your lifetime, or just like when you, like I said, when you drink milk, lactose protein might not turn on. Some of the effects that this could lead to then is making an incorrect gene product, creating a wrong amount of a gene, or creating the wrong gene product altogether. I don't know how I just turned off my camera. Okay, so you also might relate mutations to cancer. And cancer is just uncontrolled cell growth. How cancer can arise, it can be genetic changes that cause cancer can be inherited from our parents. They can also arise during a person's lifetime as a result of errors that occur as cells divide. So these errors would be referred to as mutations. Or they can also occur because of damages to DNA caused by certain environmental exposures. How we talked about UV radiation, X-ray, just like radiation in general. The video also mentioned like smoking and carcinogens, things like that. There are hundreds of types of cancers, each depending on where the uncontrolled cell growth is in the body, or where the cell growth is happening in the body. Here are some examples. So a carcinoma would be in your lung tissue. A sarcoma is just in your soft tissue of the body. And melanoma would be if you got cancer of the skin. And then you might hear the terms malignant or benign, and that's just how the cancer is growing. So malignant tumor is a cancer cell that have spread and have created tumors, and then benign tumors are a tumor with cells that are not cancerous. So it's just uncontrolled cell growth, but it is not actually causing the disease of cancer. Some external causes that we were talking about was tobacco, alcohol. We've talked about radiation. You might have heard about asbestos, like on those infomercials late at night. Some plastics have some of these carcinogens in them, and some bacteria. Obviously, it's super harmful and not common bacteria, but it is possible. And then just an overall video at the end that I want to give you, and then we'll go over the short assignment that I have for you. But it's just like a short, did you learn something about mutations? So we'll listen to the Amoeba sisters, what they have to say to wrap it all up, and then I'll go through the short assignment with you.
Ever since I was a kid, I have been curious about mutations. I think certain movies may have had an impact. I used to imagine mutations with amazing abilities, but there was also a lot I didn't understand about mutations. Let me explain. Many people understand that a mutation is a change of genetic material, more specifically, a change within a nucleic acid. RNA and DNA are both types of nucleic acids. Therefore, anything with RNA or DNA can have a mutation. That means animals, and that definitely includes humans, also plants, fungi, protists, bacteria, archaea, they can all have mutations. Additionally, so can viruses. Many mutations can be neutral in effect. You can see this codon codes for the amino acid leucine. But if it experiences this particular silent mutation, even though a base has been altered, it still codes for leucine. It did not change the amino acid. Mutations can also be harmful or helpful, but it's important to understand that mutations are random. The organism can't will itself to get a certain mutation. You can learn more about this in our natural selection video, where we talk about how a bacterium may already possess a helpful mutation that allows it to survive an antibiotic. But the bacterium didn't will itself to mutate to develop this certain mutation. Okay, so again, mutations are random. But there can be factors that make mutations more likely to occur. External factors like certain types of chemicals or excessive radiation, or internal factors like an event that causes a problem during DNA replication in interphase. During interphase, cells can replicate DNA before they divide. So let's discuss some different types of mutations. We'll start with gene mutations. DNA makes up genes, and genes can code for proteins that influence different traits. So when a mutation in DNA happens, which specifically means a change in one or more DNA bases, then different proteins can be produced, which can affect an organism's traits. In this example, we have fruit fly DNA. Mutations could include substitution, which means the wrong base is matched. Insertion, which means an extra base, or bases, are added in. There is also deletion, which means a base is removed. Insertions and deletions have the potential to be especially dangerous. Why? Remember in protein synthesis how we talked about how bases are read in threes? Well, if you add a base or remove a base, suddenly the number of bases total has changed. And if you read the bases in threes, depending on where it happened, everything that is read afterwards could be affected. We call this a frameshift mutation. If you look at this frameshift example here, where a single base has been inserted, you can see how the following codons, which have 3 bases each, are now all affected as the reading frame has been shifted. This example shows how it can lead to many amino acid changes. There are also chromosomal mutations. Remember that chromosomes are made up of DNA and protein, highly organized, and they have lots of genes on them. The human chromosome number is 46, with 23 from an egg cell and 23 from a sperm cell. This type of fruit fly here, however, has 8 chromosomes, so 4 came from an egg cell and 4 came from a sperm cell. Some examples of chromosomal mutations include duplication, where extra copies of genes are generated, deletion, where some of the genetic material breaks off, inversion, when a broken chromosome segment gets inversed, which means reversed, and put back on the chromosome, or translocation, when a fragment from one chromosome breaks off and attaches to another chromosome. There's more mutations than what we covered, of course, but the idea is that there are many different kinds of changes that can happen. If a mutation is going to happen, we already mentioned that there are especially vulnerable times, such as during DNA replication, but also there are other times too, like during meiosis. In fruit flies and other animals, meiosis makes sperm and egg cells that can have the number of chromosomes as the organism. However, sometimes those chromosomes don't separate completely. We call this non-disjunction. This can result in an egg or sperm cell that has too many or too few chromosomes. It's possible for a mutation to be passed down to offspring. Consider a protist with a mutation. Many protists reproduce asexually, and when they divide, the daughter cell can inherit the same mutation. A fruit fly, which reproduces sexually, can pass a mutation to its offspring if that mutation is found in the genetic material of the sperm or egg cell. You may wonder why we've been mentioning fruit flies so much. Well, they're awesome, but also it turns out that fruit fly mutations and how they are inherited are frequently studied. We have some links in the video description for more info. These studied mutations can also occur in humans, and we'll give a gene mutation example of a substitution in the case of sickle cell anemia. First, a little background. Hemoglobin is a protein in your red blood cells that helps you carry oxygen. But in the disorder sickle cell anemia, the gene that codes for hemoglobin is mutated. If you inherit 2 copies of this gene, one from each parent, you can have this disorder. This disorder can make it difficult for your red blood cells to carry oxygen because the shape of the red blood cell is affected from this mutated hemoglobin protein. This can lead to anemia and other problems. While unfortunately there is not a cure for this disorder yet, the good news is that treatment for this disorder has greatly improved. Another thing to mention, if an individual only inherits 1 copy of the mutated gene from one parent, they are a carrier, but they don't officially have the disease. Usually they do not have symptoms, but those that are carriers appear to have a protective factor against malaria. Malaria is a disease caused by a protist that can be transmitted by mosquitoes. These individuals can still get malaria, but their symptoms are often less severe. We should mention that studying mutations and genetic disorders is a large and important field right now. Genetic counselors work to help families that may be affected by genetic disorders. If you have an interest in learning more about careers related to this topic, please check out some of our further reading suggestions in the description below. Well, that's
Great. So that was our short lesson on mutations. Now I just want to go through the assignment. I can make it due tomorrow, like first thing, because you guys will have a half an hour to work. But I did just want to give you guys something so that I know you learned about mutations. Working. So I'm going to show you the document and work through or like tell you what you're doing. So there's 2 parts, but they're both pretty short. Part 1 is just about completing the boxes below, and you're going to classify each mutation as a deletion, insertion, or a substitution. If you don't remember or know what those mean, they're right here. So a deletion is when a base, which these are all bases, the letters are bases, same thing. So a deletion is when a base or a letter is lost. Insertion is when there's an extra base added or put into the sequence. And a substitution is when one base just gets switched for another one. So your mRNA sequence from the original DNA, you would just type things like what pairs with T in mRNA? What pairs with T? And then what pairs with A if you're doing mRNA? What goes with C? I already asked for A. Ooh. Sorry, the spacing is bothering me. Okay, and then just my last question, what pairs with a G? C. All right, so you answered all my questions. Oops. There we go. Okay, so that is the original mRNA sequence. That's what it should look like. Then when you go through your mutated DNA sequences right here, and then you're going to go through and read your mRNAs again. And since I just asked you, I will, excuse me. There we go. I will just write them. We already went through this. So you told me that it's an A and then a U and then a G. There's another A. So you just fill this out again. And look, I already found where something happened. So it says there's a T here now. So what pairs with T is an A. So if you look, that's where it switched. Because I have AUG, and then U, but then there is an A. Let's make it a red. All right, so we know that this one is the one that changed. You can highlight it red so that you know it's wrong or isn't correct. And then your question about will there likely be side effects? You can just take a guess. Like if something switched and it's completely different, do you think there will be side effects? Probably, because it's not the same sequence. It has the start codon, but then it's probably a different codon. You can just say probably if you think there is or not. And then what type of mutation is this? So we switched out. Oh, well, we should probably finish to see if it gets longer or not. Yeah, so far it would be a substitution, but let's just make sure the rest is equal. Oh no, don't highlight that part. Ah! All right, there we go. All right, so then compare the rest. This part to the top. And it all looks the same. It didn't get longer, so it wasn't an insertion because there's not an extra letter. So it's just a substitution where one base was substituted for another one. So you just write. Apparently I'm yelling substitution. So that is what you do for part 1. And then part 2, you just are going to watch a video about GMOs because GMOs are actually from a mutation. And I just want your opinion on some of these. So some of the questions are in the video, but then at the end it says like, what is one negative? What is something that you think is it good or bad? So it's only like a 4-minute video.